Let us understand the protein engineering. So what is protein engineering actually? So protein engineering is basically making proteins structurally more stable and functionally modified and active. Basically, protein engineering is associated with enzyme engineering or production of newer types of enzymes. The simple thing to understand protein engineering is there are two processes to understand this thing. So we can divide it into two types. So first type is your rational approach. And second one is your irrational approach, or to which we call directed mutagenesis, or what, evolution, directed evolution. So as we also already know that what is the central dogma of the molecular biology, that is DNA, replication, transcription, RNA, or more specifically mRNA, then RNA translate and will form protein. So if you want to change the structure of this protein, you can change the structure of the gene. Any change in the structure of the gene will eventually expressed in the changed structure of protein. So basically it is for simpler molecules, simpler proteins, but as the enzymes having three-dimensional structure, that is tertiary structure, which having coiled structure, that is, so that is post-translational modifications. So if you want to engineer an enzyme that is structurally more stable and having more affinity towards the substrate and more activity, so you have to go for the irrational approach or the directed evolution. So directed evolution is completely different approach in which we form several mutant copies of the gene that account for specific enzyme. Once we make billions or trillions of the copies of these this mutant cloned gene, and we try to express them and get the results of the result in the form of protein, and the protein, and then after this we have to go for high throw output screening. Just it is the same as that we have do for your new drug discovery thing. new drug discovery in which we takes 10,000 molecules and then lead or hit molecule we have to isolate. So this same thing is actually done in the irrational approach, and basically it is more pronounced in silico approach and computer aided approach. So this is all about the protein engineering. Thanks you all.